Hello, I am Chengxin Zhang from University of Michigan. My project is detecting gene ontology misannotations using taxon-specific weight ratio comparison. In this study, we developed a new method to find incorrect function assignments in UniProt Go annotation and analyze where these misannotations come from. Figure A shows that 99% of Go annotations for UniProt proteins are from fully automated predictions, also called infer from electronic annotation or IEA in black. Among model organisms such as human and mouse in Figure B, where experimental function annotations are more common, as shown in the white areas, there are still 54% IEA annotations in black, while another 16% are manually reviewed computational predictions in gray, such as IBA. Such a high portion of computational annotations is partly due to the exponential growth of UniProt database shown in the black curve, which has 180 million entries in 2020, while only 561,000 are manually annotated proteins in ScriptsPro shown in gray. The common usage of computational predictions in large databases inevitably introduce errors, which we aim to study. This table lists the 20 species this project focuses on, including model organisms and common pathogens. Misannotations for this species, we first classify all annotations based on different taxa of animals, bacteria, archaea, etc., as shown in different columns. Finer grain classifications, such as vertebrate versus invertebrate, are also possible. Then, for every protein function shown on each row, we calculate the portion of proteins that are annotated for this function among all proteins in this taxon, as calculated in equation 1. The annotation rate in equation 1 is illustrated as the area of the rectangle. We then compare the annotation rate among two different taxa by dividing the smaller annotation rate by the bigger annotation rate to calculate a ratio of annotation as in the next equation. The smaller the ratio, the more likely is the annotation incorrect in the taxon with smaller annotation rate. For example, the last but one function, mitochondrion, is common in animals, fungi, and plants, but very rare in bacteria. Therefore, mitochondrion in bacteria. By reviewing annotations detected through this ratio of annotation rate analysis, we find around 250 incorrect annotations among the 20 species we study, as shown for UniProt Go annotation 2018 and 2019. Although IEA is the most commonly used evidence among all annotations in UniProt in the first column, for the subset of incorrect annotations in column 2 to 3, the most common annotation method is IBA, or infer from biological ancestor, which is expert review prediction based on phylogeny analysis. We analyze why IBA is prone to misannotation, using as a case study the well-characterized UDG protein from the bacterium Bacillus in Figure A. which is incorrectly annotated with Gotham, mitochondrion, and nucleus due to over-interpretation of phylogenetic in IBA annotation process. A phylogenetic tree for the UDG family is shown in Figure B, which has two main branches. The top branch is for eukaryotes such as animals, fungi, and plants, while the lower branch is mainly for bacteria, including Bacillus circled in dashed box, even though some plants also belong to this branch. Since both the eukaryotes in the top branch and the plants in lower branch have the mitochondrion and nucleus Gotham, the IBA annotation process propagates these two terms to the most recent common ancestor, which is the root term. This causes all proteins in the family, including the bacterial proteins, being annotated with the eukaryotic function. Finally, we use Go annotations in 2018 that are later rejected or confirmed experimentally in 2019 to estimate the error rate of different types of Go annotations, confirming that IBA terms have the highest estimated error compared to all other types of Go annotations. We have reported our findings to UniPro and the Go Consortium, which hopefully address the issues in near future. More details of this study are included in a recently accepted paper, which is the first citation in the reference list. Thank you.